Hello, EAC. My name is Alexa Buger. I'm the MGAL Core Manager, and you may recognize me from previous EAC meetings. This year, I have a pilot project supported by the ICEMHH where we are investigating the effects of selenium on the gut microbiota of the mosquito vector Culex. I'm fortunate to be working with an amazing team on this project. My mentors, Dr. Matt Medeiros and Dr. Lucia Seal, are experts in mosquito model systems and selenium biology, respectively. My expertise in microbiology and isolating niche microorganisms has served me well in this role as PI for the project. And we have Braxton Ramos, an undergraduate, working with us. And she also has her own UROP funding for a project related to this one that I'll describe a little later. And now I'd like to give a little bit of background to stitch together these three seemingly disparate pillars of this project: selenium, fungi, and mosquitoes. Selenium is an essential micronutrient that is essential to host health. It is a strong antioxidant and contributes to maintaining redox homeostasis. There's a narrow range of selenium adequacy in the diet. Too little or too much is detrimental to health. There are three main biological pathways that specifically incorporate selenium into biomolecules. The incorporation of selenium into the amino acid selenocysteine is often referred to as the 21st amino acid, is the most common pathway and is utilized by all domains of life. The other two are only observed in bacteria and archaea. It's believed that bacterial transformation of inorganic to organic forms of selenium makes the micronutrient more bioavailable to the host. However, it's also known that under conditions of low intake, microbiota with first access to the nutrient can sequester it from the host as well. And it's been described that altered selenium intake can affect gut microbiota composition, colonization, function, and metabolism. Um, but um, so it's appreciated that the microbiome-selenium relationship affects host health, but the precise mechanisms as to how this occurs remains mostly unknown. About a quarter of bacterial genomes encode SEC, and interestingly, gut-associated microbiota are enriched in the ability to utilize selenium in biomolecules. Um, until recently, it was believed that fungi and land plants had lost the ability to produce selenoproteins altogether, but a recent comparative genomics analysis revealed the presence of the SEC incorporation machinery in the genomes of early branching fungi. And this order of fungi, the Harpalales, are a special group of fungi that make a living in the guts of aquatic arthropods. In this project, this is referring to the larval forms of mosquitoes. And so the Harpalales fungi symbiosis is an ancient relationship that is obligate for the survival of the fungi. And the SEC-producing ability of symbiotic gut fungi begs the question as to whether this trait is essential for the ability of the fungi to reside in the insect gut, and if it confers an advantage to the host health and survival. And we'll investigate this relationship um, using mosquitoes as a model system. And so I'll refer to the Harpalales group from now on as gut fungi for simplicity, but I'd like to describe a little more biology that makes these fungi special and unique in the gut microbiota. And as mentioned, this symbiosis is ancient. It's believed to have co-evolved with insects. The interaction is highly host-specific. Oftentimes, a new species of fungi associates with a particular species of interest. And beyond that, the location where the fungi reside in the gut is also highly site-specific. When gut fungi spores are ingested from the aquatic environment, they physically attach to the gut wall lining through a holdfast structure and secrete a material as seen down here. The entire vegetative growth stage occurs only inside of the host. Spores are released from the vegetative material, perpetuating the fungal life cycle. And so little is known about the symbiosis, even at the most basic level. Whether the relationship is commensal, benefiting only the fungi, or mutual, benefiting both the host and the fungi, is unknown. And so in this pilot project, we want to address some key surface level biology about this system. Is the SEC trait and production of selenoproteins by the fungi requisite for the symbiosis? And does production of SEC by the gut symbiote impact the host health in one direction or another? And in this project, using mosquitoes as a model system, we hope to shed light on mechanisms of selenium biology by the gut microbiota that may be relevant to human health. But we're also interested in this question from a more applied standpoint of mosquito vector control. So biocontrol methods in areas of high vector-borne disease burden rely heavily on mass rearing of mosquitoes for environmental release, and the symbiosis with this gut fungi and the microbiome in general has been largely ignored in rearing settings, and we're working to understand how proper establishment of microbiome impacts the health of mosquitoes and the overall success of rearing efforts for vector control. So to sum up the introduction, we're working to use mosquitoes, specifically the larval stages of model system, to trace microbiome host relationships with regards to selenium metabolism. We'll do this by rearing mosquito larvae in controlled mesocosms in the lab. Our first aim is to use wild diverse microbiomes to seed mesocosms and describe the ecological patterns in microbiome changes relative to selenium treatment. Key microbes that are relevant to host health and survival will be targeted for culturing. And the second aim is to create synthetic mesocosms with cultured bacterial strains, again, profile the ecological patterns with selenium treatment. This aim will enable us to dissect empirical mechanisms and microbiome level function that are relevant to altered selenium levels. And the third aim is to describe the role of this gut fungi in the system and parse out whether the colonization of the gut fungi in larval mosquitoes impacts their health under changing selenium availability. All in all, using mosquitoes as a model system, we'll dissect mechanisms relevant to microbiota selenium metabolism in the gut and the resulting impact on host health. So we have heavily utilized the InStar core for this aim, specifically for our aim one. Specifically, we relied on the skills and expertise of the InStar manager Nicole Yunaishi and the technician Ibis to learn how to work with Culex. We also used InStar incubator space and technician time for setting up and running our mesocosms. And the MGAL core is also heavily used in this project. We've done all the molecular work in MGAL. And so we've shown that we can use nutritionally defined selenium-controlled mouse chow from Dr. Steele's lab as larval food for mosquitoes. And this is significant because normal larval food contains liver powder, which is high in selenium and restricts the selenium treatment conditions. And we've run 2 separate mesocosm experiments so far, and I'm marking these as a challenge only because we still need to repeat them and iron out all the wrinkles. But nonetheless, we were able to collect material for DNA extractions and library preps, and we anticipate having the data back sometime in April. We were also able to use total X-ray reflective spectroscopy, or TXRF, technology to quantify total selenium in the larvae and in the housing water. And this equipment is housed in Dr. Steele's lab, and it was acquired recently by the Precision Nutrition COBRE, another COBRE in our department right now. And so we've tested and optimized techniques for TXRF analysis of larvae and developed a technique to process a single larvae in parallel for selenium quantification and microbiome profiling. So since I don't have the sequence data back yet, I'll just like to share some pictures and describe our process. We identified this pool, as I mentioned, that was teeming with mosquitoes, and it was essential to the success of our project, as you'll see later. And while we were elated about this resource, the people in the area were not so much. And so in the end, not too surprisingly, the pond was poisoned by the landscaping crew, but for months before that, we could come to this pond and we could collect everything we needed. That we could collect larvae, water, and egg rafts from the single source right on campus. And we also collected some stream water from Manoa Stream to serve as an inoculum for our wild mesocosms. So in the first experiment, we set up mesocosms testing the 2 sources of wild inoculum, the kiddie pool water and the Manoa Stream water. And the treatments were the food, the sufficient or the high selenium level mouse chow. And mouse chow is colored blue for sufficient and red for high selenium, and that in combination with the brown pond water gave all these different rainbow of colors to the mesocosms. We used 100% source water that had been strained at 100 microns as the source, inoculum source, and we used crushed food. And the combination of these 2 factors led to bacterial overgrowth, and we had some issues with biofilm, which impacted the survival of the larvae. But nonetheless, we were able to collect in usable material from this experiment, and you can see that the larvae did survive and grow well compared to the control, or they grew, the ones that did survive grew well compared to the control condition. So we repeated the wild mesocosms a few weeks later. This time we weighed food in a single chunk, which significantly reduced the turbidity, and we used only 1 to 1,000 inoculum source instead of 100% source water. And so we didn't have any issues with biofilm growth in the mesocosms, but unfortunately the egg rafts that we collected for the experiment had come after the pond had been poisoned, and so none of the larvae survived. And so we'll have to repeat this one again using these optimized conditions. And so we repeated, or for aim two, I don't have a lot to report right now, as these experiments are just on the horizon. We do have material preserved in glycerol for isolation of bacteria from the material harvested from experiment one, and we're fortunate to be working with Mamo, Dr. Medeiros' grad student, who also works with Culex mesocosms, and Mamo already has a collection of about 12 bacteria that yield healthy mosquitoes that we intend to piggyback on her efforts setting up mesocosms to run our experiments with selenium treatment, and this is planned for April. And so now I'm very pleased to share with you our progress for aim 3 and let you know that we have successfully isolated and are continually culturing our Harpalales gut fungi that we isolated from a larvae that we collected from the kiddie pool. And we are situated to start introducing this fungi to mesocosms and begin working with the fungi genetically to dissect host-related mechanisms that are relative to selenium metabolism. And I'm showing here in these pictures the process of isolation, which is slightly unlike any microbiology I have ever done before. We carefully dissected guts and we could visually identify the fungi in the guts of the mosquito. And we had some issues with penicillium overgrowth. We struggled with this issue for about a month. But finally, after perseverance and hundreds of larvae dissections, we found a larva that was amenable to isolation of the gut fungi. And so here is our isolate growing in pure culture and showing this beautiful branching morphology in apical trichospores. We're able to collect these trichospores, which will be used for inoculating our mesocosms with the gut fungi. And we haven't sequenced the fungi yet, but we did run a PCR with Harpalales specific primers for verification that we had what we were looking for. And so now we're ready to proceed with adding the fungi to synthetic mesocosms, and we're planning on including these treatments to our experiments planned with Mama this month. So in summary, we're building synthetic mesocosms with and without the gut fungi present, and we're using selenium as a treatment with the mouse chow delivery. We are improving our TXRF data collection, analysis, and standardization for selenium quantification of the material from the mesocosms, and we're analyzing our sequence data from the first experiment. On the horizon, we're sequencing the fungi genome, we're building synthetic mesocosms, including the gut fungi, and we're repeating the wild mesocosms with the fresh material, and we're working to isolate bacteria that are relevant to selenium treatment. And we have not experienced too many bottlenecks besides the kiddie pool becoming unusable. We do have to wait a bit for the one MySeq available at UH Manoa, so our sequencing data has been a bit delayed. And Instar has had some issues producing enough egg grafts for the lab from their lab-reared Culex, and access to wild sources with accessible egg graft collection is limited, as you saw with the pond. But these issues are being readily addressed by Dr. Medeiros' group, who maintains their own buckets for wild mosquito collection, so I don't anticipate this being a significant issue as we continue working with Mamo. So now finally, I'll share some of the products that we have so far. Although unrelated to this project, I did publish a paper since starting. It was published last September in Jove, and it's relevant to the work I did as a PhD student working with the micronutrient zinc on mycobacterial physiology. In April, Braxton will present the results of her research at two separate venues at UHM, the CTAR Symposium and the Undergraduate Showcase. Braxton has focused on co-opting the Harpalales-specific primers to develop a qPCR assay to investigate the prevalence of the gut fungi in larvae from the wild. And just in this last week, I was connected with the Hawaii State Vector Control Lab, who collect mosquito larvae across the state. And I'm actively pursuing this collaboration, and we hope to significantly expand the scope of this aspect of the project through this newfound avenue. That's exciting. Through the suggestion of Dr. Seal, I plan to attend a major selenium conference that unites the global selenium biology community held once every 3 years, and this will be this year in October in Korea. And finally, I do anticipate writing at least one grant this year that is relative to this project. In collaboration with Dr. Medeiros, we want to bring the gut fungi to mosquito rearing systems and specifically address the microbiome function in the lab setting versus the wild. And we have not identified a specific funding mechanism, but I think the NIH notice from NIGMS that's focused on understanding microbiomes in the built environment is a good fit for this work. I also hope we can write another grant specifically investigating the role of SEC production by the gut fungi and its impact on the symbiotic relationship, and this approach will focus on the role of selenium and its exchange between the fungi and the host. And finally, I have another project that I've been actively pursuing with my industry collaborator in the aquaculture space for about 3 years, which utilizes a niche marine eukaryote that I've isolated from nearshore Hawaiian waters, and we're resubmitting our NSF SBIR grant to this end. And thank you so much for listening. I'm excited about this project and what's to come, and I'm looking forward to seeing you at our meeting in a couple weeks. Aloha.